Hi, everybody. We're going to continue with our skeletal muscle lecture today. And we're going to kind of review some of the topics I discussed with you guys in the last part of the lecture, and then build on that in terms of muscle contraction. Quickly reviewing what we discussed previously, we talked about how myosin and actin are interacting with each other. That basically leads to muscle contraction. So in this picture, if we are looking at our main components that are involved in muscle contraction, you have your myosin, which are these basically structures right here, and these extensions coming off of them. These are your myosin heads, right? And down here, what we have is my actin, and the little indentation inside of this would be the myosin binding sites, which we discussed last time, but again, I'm just kind of depicting it on a picture for you today. So under normal condition, what we have is what we want to have for muscle contraction is these myosin heads to interact with these myosin binding site on the actin. And again, kind of like the tug of war, these are gonna move in and they're gonna pull the actin in that direction, basically pull actin inside the myosin. Now, we also talked about the fact that when we are when you are dealing with muscle relaxation, as in the absence of the electrical impulses from your nervous system, I do not want the actin and myosin to be interacting with each other. So for that, I use two regulatory proteins. The one that is literally covering the myosin binding site and is depicted as this line. These are called the tropomyosin. And these little circles that I've drawn, these are subunits of a protein called troponin. So troponin and tropomyosin together, what we call these two, are regulatory proteins. And again, the goal of these regulatory proteins is to prevent interactions between actin and myosin when your muscle is relaxed. Now, when muscle is going to contract, one of the main things that happen, and I'll discuss that again in more detail later on, is that the calcium that was stored inside the sarcoplasmic reticulum is released. So when that calcium, if you guys remember calcium, we talked about it last time, stored inside the structure called sarcoplasmic reticulum. So when that calcium released and it comes in this environment, right? Calcium goes and binds to your troponin. So let me do my colors. So calcium comes and binds to my troponin, and I can draw it actually up here because it makes it a little bit easier. So calcium comes in and binds directly to my troponin. So the interaction between calcium and troponin causes the troponin to move. Now notice how troponin is connected to the tropomyosin. So if I'm pulling or moving the troponin from that site, it's also going to pull away the tropomyosin. So calcium connects the troponin, moves the troponin out, and with it you're pulling or moving your tropomyosin. That allows what? The myosin binding site on the actin to free up, which is what I depict here. And therefore, in that scenarios, these interactions between the actin and myosin heads can take place, and that causes the muscle contraction. So if we're talking about the importance of calcium up to this point, what you know is the calcium that is stored inside the sarcoplasmic reticulum is essential for creating muscle contraction by removing the regulatory proteins and allowing the actin and myosin to interact with each other. Now, building on what we talked about last time also, we talked about muscle contraction. We said muscle contraction depends on the nervous system, more specifically the category of cells called neurons or nerve cells. These neurons or nerve cells create electrical impulses. that causes electrical impulse inside my muscle fiber, and that electrical impulse leads to muscle contraction. We also take a look at the structure of the neuron. We said neuron is made up of three main parts. Dendrites, which are receiving electrical impulse from whatever was the upstream from that, so receiving the stimuli, and pass that stimuli to your cell body or soma. Cell body importance is that it is creating a specific type of molecule called neurotransmitter because it has all of the organelles and nucleus needed to basically accomplish the task. Now, these neurotransmitters are eventually gets transported into the end of your neuron called axon terminal. And in terms of electricity, electricity starts with the dendrite, goes to the cell body, goes all the way through the axon and eventually into the axon terminal. Now, why do I care about the neurotransmitter that is produced by the cell body and transported to the axon terminal? It is the way for my neuron to communicate with the skeletal muscle. It is a way for the electrical impulse to be transferred from the neuron into the skeletal muscle fiber. So let's take a look at more detail about this. Now, looking at a neuron and the skeletal muscle, which is what is depicted here. Here is your axon coming down. End of the axon is the kind of enlargements. We're only showing or depicting one of them, but there are many. This enlargement at the end is called the axon terminal. And within the axon terminal, we have these little tiny vesicles or pouches that contain my neurotransmitters. Again, I just mentioned earlier that neurotransmitter is a means for me to communicate with my skeletal muscle fiber. Now, the specific neurotransmitter that is produced and stored by your neuron in case of muscle contraction is called acetylcholine or ACH. So if you see these three letters, automatically think about neurotransmitters utilized during muscle contraction. Now, what I've done for you guys is I've depicted this in a very simplified picture to kind of highlight how everything we discussed so far come together to cause muscle contraction. So you're welcome to recreate this for yourself. I'm going to talk about it briefly. Again, this is not as detailed as you will see in physiology. It is to just kind of give you a generalized idea about muscle contraction. But basically what you have is electrical impulses. So let me label the structures for you and then we'll go from there. So what we have is your axon. Make sure it's black. The end of your axon is axon terminal, and try to kind of follow this and try to say it as I'm saying the name as well. These little circles are your vesicles with neurotransmitter. NT is neurotransmitter, acetylcholine, which is shorthand to ACH. Now, what we have down here is my muscle fiber, right? This is a skeletal muscle fiber. Again, the membrane that is for your muscle is sarcolemma. And the space inside my muscle fiber is called sarcoplasm. Now, what we have is the continuation of the sarcolemma or indentation, invaginations of the sarcolemma into my muscle fiber, which we mentioned earlier is essential for a fast spread of electrical impulse through the muscle fiber. If you recall, these structures are called T-tubules. And T-tubules basically wrap themselves around or are present around these structures called sarcoplasmic reticulum. Sarcoplasmic reticulum, and sarcoplasmic reticulum has a very specialized function, as it stores lots and lots of calcium inside of it. And again, we already have a clue that this calcium is needed for muscle contraction. Some of the other structures we have here, based on the previous drawing, you can tell that this is my myosin. These little extensions are my myosin heads. Again, please try to say it with me to kind of help you guys practice the identification. What we have here, these are my actin. The little spaces inside of them are my myosin binding site. These little circles right here are my troponin, and the long extension is my tropomyosin. So, few things that are new for you guys. One might be wondering what they are. What we have here is that, as you can see, the neuron, which is up here, is not directly coming into contact with my muscle fiber. There is a gap between them. This gap is called the synaptic cleft. This is a synaptic cleft. This is where neurotransmitter is released into. So this is a way basically a neuron communicate with the muscle. You will have your little dots of neurotransmitter inside of here. So if these are my neurotransmitter, right, they can get released into this space right here called the synaptic cleft. Now notice that these neurotransmitter can interact with what I've drawn here as these boxes. What these boxes depict are a specific type of channels called sodium potassium channels, and they are actually going to interact with ACH, and when that ACH interacts with the channels open up, allowing the ions to move in and out of my cell. Following this, what I also didn't talk about is these little boxes right here. These are also proteins that are allowing this calcium that is stored here to be released into my sarcoplasm. So these boxes are basically proteins. They have very specific name, and the name kind of tells you their function. They're called calcium voltage gated channels, which basically means they are only and only going to be activated when there is an electrical impulse passing through my muscle fiber. So again, this is a simplified version of what you will learn in terms of the physiology and muscle contraction. Last word before we look at the brief overview of this process is the area where a neuron is communicating with my muscle fibers. So this region right here, where that communication happens, look at the name, break it down for yourself, called neuromuscular junction. This is a space basically, or the area where a neuron is communicating with the muscle, and that communication is through the neurotransmitter ACH. So let me walk you guys through this step by step, and again, kind of giving you an idea. I do expect you guys to have at least the understanding that I'm describing here. Please, again, I keep reminding you guys this. This is a simplified version of what you will learn in more detail later on. So what we have is electrical impulses coming down the axon. The electrical impulses reaches the axon terminal and causes the vesicles of the neurotransmitter ACH to be released into the space called the synaptic cleft. These neurotransmitters can interact with my protein channel called sodium-potassium channels, which allows, through this interaction, sodium to get out and potassium to come in. And that creates these ion movements, basically create what we identify as electrical impulses. So which is this arrow that I'm depicting. This is my electrical impulse. Again, this is a simplified version. This is more complex than this, but for us, this is all you need to know. Now, this electrical impulse needs to be spread through the muscle fiber. So it's gonna go down the T-tubules. And these electrical impulse activate these channels, which are your calcium voltage gated channels. So what does that imply? The calcium can get out into the sarcoplasm. Calcium is free. So what can it do? Calcium can go and interact with my troponin and move my troponin. And remember, if troponin moves, tropomyosin also moves with it, which means that now my myosin binding site is free. I can interact with my myosin heads. And through that interaction, we get what? Muscle contraction. Please do not make this more complicated than it is. It is a relatively simple procedure that is done in terms of, or what you need to understand at least right now. Don't make it more complicated. So let's take a look at it in a picture, guys. Again, highlighting some of the important concepts we talked about. Here's my axon terminal receiving electrical impulses from the axon. So here's my axon terminal. These little circles that you see are vesicles that contain neurotransmitter, ACH. Notice that they're dumping their material into this gap right here, which we identify as the synaptic cleft. Notice that ACH is binding to these channels you see in purple. So you already learned that these channels are called sodium-potassium channels. So remember those. Same picture again. So neurotransmitter is being released. Follow the arrows, guys. Neurotransmitter creates the electrical impulse. Electrical impulse goes down the T-tubules, which is the structure you see right at the center. That causes the calcium, which you're seeing right here, to be released inside the muscle fiber. And again, that calcium is what allows, based on our picture, moving the troponin and tropomyosin in turn, and allowing myosin and actin to interact with each other, causing muscle contraction. So that's just as far as we're gonna go in terms of muscle contraction. One last thing actually before I finish up is how you go from contraction to relaxation. One of the main ways that you do that is that there are enzymes that come into the synaptic cleft and they break down your neurotransmitter ACH. And as they break it down, basically, ACH, if you remove the ACH basically from here, so the enzyme come here and break here down the ACH. So sodium-potassium channels are closing up again, which means no electrical impulse. That scenario, the calcium that was released into the sarcoplasmic reticulum gets reuptaken by the sarcoplasmic reticulum again. This is an active process, which means it needs energy. And again, if there is no calcium, troponin and tropomyosin are going to prevent the myosin-actin interaction, and that causes the muscle to relax back to normal. Now, another concept we want to talk about is in terms of what we identify as motor unit. You need to understand that you cannot have a 1-to-1 ratio for neurons communicating with individual muscle fibers. So you have literally millions of muscle fibers, which basically makes it very difficult to have one neuron for each of the muscle fibers. So what you have in reality is you have the axon of a neuron that comes in, branches and form many axon terminals, as you can see right here. So here is your axon, here are multiple axon terminals. And each of those axon terminals are communicating with the muscle fiber. So they form multiple neuromuscular junctions with multiple muscle fibers. So when we're talking about motor unit, in reality, the definition of motor unit is the neuron, one single neuron, and all of the muscle fibers they innervate. So again, this is not a one-to-one ratio. You have one neuron, but many muscle fibers. Now, in terms of looking back at our muscles, we learned that the smallest structure that makes the muscle is your actin and myosin. And again, this is a really good review, so try to say it with me if you can recall it. Actin and myosin are my myofilaments that are packed inside our structures called sarcomeres. Sarcomeres are packed inside myofibrils or arranged in myofibrils. Myofibrils are found inside my muscle fiber. A group of muscle fibers make my fascicle, and many fascicles put together make my whole muscle. Now here in this slide, you notice that it says fascicle orientation. So how does the fascicles that are basically form my muscle can arrange themselves? And there are 5 different ways of arranging themselves to make my whole muscle. They can form what we identify as fusiform, parallel, convergent, pennate, or circular. Some of the names are pretty self-explanatory. For instance, fusiform implies that the muscles are going to be thick at the center and narrow down on either end. These type of muscles are typically or moderately strong in terms of contraction. And some of the most common examples of these type of muscles are either biceps brachii, which is your arm muscles, or gastrocnemius, which is your calf muscle. Parallel muscles, as the name implies, is more strap-like, the same level of thickness throughout the muscle. They are contract more than other muscle type. However, they are weaker than the fusiform that this would describe. Some of the best examples of the parallel muscles are rectus abdominis. Typically, people call this your six-pack, quote unquote. So these are the muscles that form the mid portion of the abdominal muscle. Sartorius muscle is another muscle. This is on the anterior of your thigh and basically covers your quadriceps or runs over your quadriceps. Pennate muscles are feather shaped. Basically what it implies is that the fascicles is inserts into the tendon that runs right in the middle of the muscle. So you can have a single tendon that runs in the middle. That's called unipennate. And the best example of that is semimembranosus, which is a muscle that forms part of your hamstring muscles in the back of your thigh. You can have bipennate, which is your rectus femoris. This is one of the major muscles of your quadriceps, which is again anterior aspect of your thigh. And then you can have multipennate, which is deltoid, as you can show in the picture. You can see in the picture. Deltoid muscle is the triangular muscle that forms your shoulder, basically, or covers your shoulder. Circular muscles is pretty self-explanatory. So these have a circular pattern to them, and the type of muscles that are identified as circular are sphincters. A couple of examples of that, even though I only wrote one of them, is orbicularis oris, which basically is a circular muscle around your mouth. You have a similar type of muscle around your eye called the orbicularis oculi. And convergent muscle, basically what you have is kind of a fan shape. You have a broad side where it, as it comes into the opposite side, it narrows down at the point of insertion. best example for this muscle is your pectoralis major, which is the largest chest muscle we have. And basically, it is identified as one of the strongest category of muscles we have. I have already talked about this. You're more than welcome to look at the picture and review how the whole concept of sliding filament causes muscle contraction and shortening of the sarcomere. The point you want to really look at, again, is the light and dark band here versus this picture. Now, the next concept we're going to talk about is your muscle attachment. Muscle attachment, if you typically, you're talking about a muscle, you will have 2 points of attachment. One that is, in our case, will be identified as origin, and the other one as insertion. Now, origin basically is where the point of attachment is less movable during the movement of the muscle. An insertion would be the more movable attachment. So what does that exactly mean? Let's take a look at a picture of a muscle. So the muscle you see right here is a muscle called brachialis. So it starts from the humerus and it connects to your ulna. So here is a point of attachment up here. Here's a point of attachment here. So brachialis helps me flex my arm. So when I flex my arm, humerus stays relatively stationary, which basically means that is the point of origin. And ulna actually does the movement, right? And therefore, because it's the movable part of the muscle attachment, this is known as the insertion. Now, how are the insertions different? You can have insertions that are identified as direct. These are fleshy attachments where the muscle surface is attached directly to the periosteum of the bone. If you remember, periosteum basically was the connective tissue that was wrapped around the bone. And you can see it on this picture as the direct attachment right here. Indirect attachment is you have the connective tissue form a tendon, as we depict, which is basically your epimysium forming the tendon, or another structure which is more broad called aponeurosis. So tendon, again, is more cord-like shape. As you can see in this picture, it kind of narrows down and converges into one point, versus aponeurosis is very wide and thin. Best example of aponeurosis is what covers the anterior surface of the, or superior surface of the skull, holding the muscle or two parts of the muscle from the frontal bone and the occipital bone together. These muscles are called frontalis and occipitalis, held by an aponeurosis. Now, we already talked about some of these when we looked at the skeletal system. Basically, the mode markings like tubercle, tuberosity, crest, trochanters, all of these basically bumps or grooves or roughened surface of the bones are points of attachments for the muscles. And again, these attachments could either be direct or indirect. Lastly, we're going to talk about how we actually name the muscles. So muscles are named based on their location, size, how the fibers are running, how does the fascicles are running, how many origins they have for the muscle, and what is the shape of those muscles. So let's just start with something simple. Location. If you hear the word tibialis anterior, you kind of think about your tibia, the bone in your leg, and anterior as an in front. So this muscle, which you can see depicted here, is literally going to be sitting in front of the tibia, hence the location. We can categorize the muscles based on their size, such as maximum, and minimum, longest and brevis, major and minor. A few examples for that, even though it's not written here. We have pectoralis major and pectoralis minor. We have gluteus maximus, you have gluteus medius. You have flexor carpi, extensor carpi radialis longus and extensor carpi radialis brevis, which basically means long and short. You can categorize muscles based on the direction of the fibers and fascicles. For instance, if they are straight, we refer to these as rectus, like rectus abdominis muscle. The muscles are on the long axis, parallel on the long axis. Transverse, the muscles create a horizontal pattern. Best example is the innermost muscle lining for the abdominal cavity called the transverse abdominis. And oblique, these are coming at an angle. And best example again is within your or associated with the abdominal muscles, 2 layers of external and internal obliques. Number of origins, if for instance you have 2 origins, you can call them biceps, like biceps femoris or biceps brachii, and you can have sometimes 3 origins, like your triceps brachii. And then lastly, your shape of the muscles, things like deltoid muscle. The word deltoid implies triangle. The actual muscle, if you take a look at it, is a triangular muscle over your shoulder. And serratus means sawtooth, or basically that creating that pattern you see right here, seesaw pattern. So this is, for instance, serratus anterior. So basically this is both the location as well as their pattern. Okay, sorry, I guess I have one more topic to talk about. Muscle type, there are components that can help us identify the muscle fibers. These components is how does your muscle create its energy? Is it using the aerobic process, which basically means oxygen present, O2 present? Or is it using an anaerobic process, which means O2 is absent, and it's using a process called glycolysis? So if you're using O2, we call that oxidative fibers, and if you're using glycolysis, we call that glycolytic fibers. Second part of this statement, how quickly they're going to contract. So basically, you're going to have either a slow or fast contraction. Do keep in mind that if you're slow, you basically don't use as much energy, versus fast, you use a lot of energy. So if your energy access is low, then that muscle is going to get tired fast. A couple other things that help you understand the concept as we go through the PowerPoint is if you need O2, so using aerobic reaction, you're going to have high capillaries. You're going to have a significant amount of blood vessels, basically, right? That basically means blood vessels. You produce lots of ATP, so high ATP or high energy. But if you're doing it in absence of oxygen, you don't really need a lot of capillaries or blood vessels to transport that oxygen, and the amount of ATP you're going to produce are relatively small. So based on those characteristics listed, you have 3 categories of muscle fibers: a slow oxidative, fast glycolytic, fast oxidative. Let's try to understand it without even going into that information, some of the general concept. A slow means it's going to contract slowly. Fast, it means it's going to contract rapidly. Lots of energy versus it basically, it doesn't create a lot of contractile force. It creates a lot of contractile force in a short burst. Oxidative implies usage of oxygen versus absence of oxygen and utilization of glycolysis. These type of muscles are going to have lots of blood vessels. These will not have as much. And if you combine these 2 together, fast oxidative means it's going to produce a lot of contractile force in a short time. But because it has access to a lot of oxygen, it might not be tired as fast as your fast glycolytic fibers. So let's take a look. A slow oxidative is red due to a significant amount of blood vessels and myoglobin. If you're unfamiliar with that, myoglobin stores O2 in muscle. It obtains its energy through aerobic process, which kind of makes sense. It has lots of mitochondria because mitochondria is the organelle for ATP production. It is rich in blood supply, so capillaries. Again, it contracts slower and its resistance to fatigue. And the muscle diameters, the diameters of these muscle fibers are relatively small. Fast glycolytic, little myoglobin and few mitochondria, because again, basically no need for O2 and no need for mitochondria to produce a significant amount of ATP. They are typically larger in diameter, typically twice as large as the slow oxidative muscles. They contain lots of myofilaments, so a lot of actin and myosin, which creates a lot of power, but they're gonna get what? Contract rapidly and quickly get tired. Intermediate filaments are intermediate or fast oxidative fibers are ideal one. They're between the other two in terms of the strength. They do contract quickly because they are fast, like your glycolytic fibers, but because they are oxygen dependent, hence the word oxidative, they are somewhat fatigue resistant. And again, kind of think about it, because they have access to what? To lots of ATP, because they're relying on what? The aerobic process, lots of myoglobin, and rich blood supply through capillaries. And this is important if you're looking at the type of muscles. Fast oxidative are the ones that typically do your day-to-day activity. You don't want them to get tired really fast. Fast glycolytic muscles are the ones that are basically get short bursts. So if you're doing, let's say, examples like weightlifting, right? 100-meter run, right? Fast, you go through a burst and you're done, basically. These are your fast glycolytic. And a slow oxidative, best examples would be the muscles that help you with your posture, where they need to continuously contract, but they need to do that over a long period of time. So they can basically, there must be resistance to fatigue. And I think that's about it. So we're done with our muscular system. We'll talk to you guys for our next PowerPoint. So keep it up.